So I'd just like to start by saying what an honour it is to have been selected for the IUIS Rising Stars programme. And here I'm just going to give you a brief taster of the research programme that we're working on in Manchester. So what I'm really interested in understanding is how we fuel our immune responses to infection, and how our immune responses themselves control our energy balance. So as I'm sure that we can all appreciate, when we have acute infections, a viral infection, flu for example, this drives sickness-induced anorexia, where we develop this aversion to food. And yet we need to mount an immune response which is energetically demanding, meaning that we need to rely on our endogenous energy stores to fuel these immune responses. And this is fine in the short term because we normally have enough fat, enough adipose tissue and muscle to fuel these responses. But what happens during chronic infection when we've depleted these stores? So to answer this, I'm working using a model called human African trypanosomiasis, or more commonly known as sleeping sickness. And this disease is endemic to countries in sub-Saharan Africa, and it's caused by these parasites, these extracellular parasites that you can see in purple in the histology slice, which when we have chronic infection with these, they cause neurological disorders such as sleeping disorders, and they also lead to extensive weight loss and tissue wasting, which can really be exemplified here in these images. So these parasites, they infect an awful lot of different species, and regardless of the species that they infect, they induce this weight loss and adipose tissue wasting phenotype. So we decided that we would investigate this more using a mouse model. And so what we do is we take black 6 mice and we infect them for around 25 days to induce chronic infection. And what you can see nicely in the graph in the top right is that over the course of infection, the mice lose a significant amount of body weight. What you can also see is that in the bottom right is that the mice also lose a significant amount of adipose tissue mass, raising the question of what drives this weight loss and adipose tissue wasting. Well, to cut a long story short, what we found was that in the adipose tissue of mice during infection, that we have replicating T cells, not surprisingly, and what we found was that within this subset of T cells that we have IL-17 producing Th17s and V-gamma-6 gamma delta T cells. And this is really interesting to us because we know now that IL-17 is involved in controlling adipose tissue homeostasis. So because the IL-17 was coming from multiple sources, we decided to infect mice with a global deletion of IL-17AF. And what we found by doing that was that these mice no longer lose weight compared with wild-type controls. We also found that these mice retain more of their adipose tissue than the wild-type controls as well. What was also quite exciting was that when we deleted the IL-17 receptor A from adipocytes within the adipose tissue, that these mice are less able to control the parasite numbers in their adipose tissue. So you can see in the histology on the left, in the brown, we have the parasite staining, and you can see that in the knockout mice on the bottom, that there's more of this staining and more parasites. And when we quantify this using qPCR, we can also see via this that there is almost a doubling of the parasite numbers, suggesting that IL-17 through the adipocytes is essential for the local immune response, and also suggesting that the adipocytes themselves form immune hubs that help to coordinate the local immune response. So if you'd like to find out more about this, please come along to my session on the 18th of August, and I look forward to seeing you there.